Dear QUAREP fellows, dear microscopy enthusiasts, my name is Arne Seitz. I'm heading the bioimaging and optics platform at EPFL. And today I want to report on the activities of Workgroup 6, stage and focus of the QUAREP initiative. It's important that everything that will be reported is a joint effort of all Workgroup 6 members. I want to start with reporting or with outlining the aim and the mission of that workgroup because it might not be familiar to everybody. And then I want to give a summary of the recent activities, and all of the activities, and particularly the protocols I will report in, has been established throughout the regular monthly meetings. At the end, I just want to give an outlook, and I want to give you an idea of what you can expect in the upcoming year. I want to start with the mission of the Workgroup 6, and the mission of Workgroup 6 is to develop protocols which can be taught on optomechanical components, which are typically attached to a microscope in order to move the stage in the three directions, X, Y, and Z. Now it is clear that the manufacturers have their own protocols before they deliver the devices, or to send back the devices, and they will check the performance of these devices before leaving the factory. However, up to now, there are no protocols available which can be easily performed in the field. And in the field means when attached to a microscope. So the idea of the workgroup was to come up with such protocols. And it is important that these protocols are easy to perform. So they should be done by the users, and they should be report on the relevant parameters. Therefore, it is so important that it is a joint effort between manufacturers on one side and users on the other side, because we need the input from the manufacturers on one hand, and we need to input from the researchers on the other hand in order to really identify what are the relevant parameters. Before doing so, we also need to find suitable test samples, and this is also one of the ongoing efforts of the working group, because we need to find samples which are available. So we want to have something which is readily available so that it can be applied by as many scientists as possible. We want to have samples which have a long life expectancy, so we don't want to throw away the sample to one usage, and we want to have robust samples, and we want to minimize the costs. So we want to come up with samples where the costs are not prohibitive in performing these protocols. When starting developing protocols, one thing became immediately clear, and that is that temperature is a really important parameter which we need to control. What you see here is an experiment where 4 micrometer beads were recorded over 15 hours on a microscope, and at the beginning of the experiment, the temperature was shifted from 26 to 37 degrees. And what you can see in the two projections here is that the beads, they are moving along the XY axis as well as along the Z axis. And the challenge when you want to report on the performance of devices which move the sample in X, Y, and then Z is that any measurement needs to be decoupled from any deviation or any contribution which is coming from temperature drift or temperature changes. So therefore, it is very important, and this became also clear throughout the development of the protocols, that the temperature is controlled and that we have a stable temperature on one hand, and that the system itself is in equilibrium with the room temperature. So whenever you want to perform such protocols, you need to make sure that the system is switched on at least 1 or 2 hours before doing such a measurement. And additionally, you need to avoid temperature changes in the room because they might badly influence the outcome of the parameter. So we came up with 2 protocols within the previous years, and you can see that they have been already published on protocols.io, and we also got feedback from the community. So we came up with a protocol which can measure the reproducibility of the XY positioning system of a microscope. Here we just want to ensure that the devices, so that the motorized stages which are moving the sample, that they are in spec, and that we can expect that we're going back to the same position, at least what can be expected from the tolerances of these stages. And we came up with a protocol which can report exactly on this parameter. And I'm inviting the QUAREP community to apply this protocol and to give feedback so that we can further optimize the protocol. In addition, we have also developed a protocol that can report on the drift measurement of the system. This is also important in order to have a protocol at hand which can report when can we expect that the system is stable. So if you see, for example, if you recall the video which I showed you a few minutes back, then you saw the movement of the beads, and this is exactly such a drift measurement protocol. And we're also coming up with a drift measurement protocol which can be used in order to report on the stability of the system. And this drift measurement protocol can be used in order to record the drift of the stages, or if it is ensured that the stage is not drifting, then it can also be used in order to measure the room conditions. So whenever you see temperature changes, then this will directly be reflected in these drift measurements as well. These were the two protocols which were developed within the last year, and they are, as I said, they are released in protocols.io. And we definitely want to collect feedback from the community. So we want you to test these protocols and report us back what is working and what is not working. That's one thing. And the other thing is that we want to collect the results from the community. So ideally, we want to, if these protocols are applied, so then we want to have tables, or then we want to have a database which can report. So what can you expect from the stages and what when you will perform these measurements. In the future, we want to come up with a protocol which can also report on the Z movement, also the Z readability and reproducibility. It's something which is important, for example, when it is acquiring high quality Z stacks. And we want to come up with a protocol which can report also on that parameter on the microscope. And then we want to come up with an easy and simple system that can measure the temperature of the microscopic body. As I mentioned before, temperature is an extremely important issue. And ideally, we want to couple the measurement of the performance of the stage and the positioning system with we want to couple it with a measurement of the temperature, and also there are ideas in the work group how that can be realized in an easy and reliable measure. And last but not least, we want to come up with new test samples. So far we're using beads, and that is working fine, but maybe beads are not the most optimal and most suited test samples. So therefore there will be also ongoing efforts in that direction, so that we're coming up with new test samples. This is what I wanted to tell you about the activities of work group 6. As I said, this is a joint effort of all work group 6 members. In particular, I wanted to highlight the contribution on the company side from Arne Brune, Stuart Jarvis, Keith van der Oort, Roman Dantel, and the co-chair of the work group, Stan Swartz. And on the academic side, these are Claire Mitchell, Hans Fried, Martin Spitaler, and Roland Litschke. And everybody who's interested in joining the work group is invited to join us. And we're looking feedback for your comments and suggestions.